All right, so up here, this document right here, we're going to go through and draw what is happening during all the stages of meiosis. So we're going to begin with prophase. So the stages that are going on through this process are the exact same stages that happen during mitosis, right? The whole thing is going to go through prophase, metaphase, anaphase, and telophase. All of those steps and stages, the same events occur during meiosis as they do during mitosis. But there are a few differences between the different phases, and so I'm going to highlight those as we go through this drawing. So for prophase 1, we're going to have a cell that is going to have two sets of homologous pairs. So if we have two sets of homologous pairs, how many chromosomes do we have overall?
4.
So we have 4 chromosomes overall, right? So if we're looking at them this way, and they are doubled like this, where they form this X-shaped structure, what do we call this X-shaped structure? This is still called a chromosome, all right? But it's a chromosome with 2 copies that are the same. So what can we call each of these individual legs? Chromatids, right? So these are sister chromatids. So for the sake of what we're going to do here today, we'll say that anything that's in red, that's the set of chromosomes that comes from the mom. Right? And anything that's in blue, we'll say that's the set that comes from the dad. So during prophase 1, right, if we're in prophase 1, we are in what we call meiosis 1, because there are 2 divisions that happen. The goal of meiosis 1 is to separate homologous pairs. So if we are separating homologous pairs, what's going to happen to our ploidy? Are we going to be ending up as diploid, or are we going to be ending up as haploid? So we're going to be haploid, right? To be diploid, you have to have 2 sets. If we are separating the homologous pairs, we are separating sets. If we separate our sets, then we are going to end up as haploid, right? We're going to be making haploid cells. What's the other term for haploid cells? What do we call those? Gametes, right? We're making gametes with meiosis. Okay, so we are going to our haploid cells, we are going to be making these gametes. All right, so let's go up here to prophase 1 and then follow on to our next stage, which is metaphase. So I'm going to draw our chromosomes. Okay, so we have 2 sets, and I'm drawing them at different sizes just because I want to differentiate between them. Okay, so let's say we have our small set, right? That's one type of chromosome, and then this big one is the larger one. Okay. So during metaphase, what happens during metaphase? What happens during any metaphase event during cell division? So they're going to line up in the middle. Okay, so our chromosomes have lined up, but in this case, it's not the chromatids that are lining up, it's the homologous pair. The homologous pair has lined up on this point, so they've lined up on the middle. Okay. So other things that have happened, right, we have our centrioles that have formed during prophase, right, so they have started to move and migrate. So if they are in metaphase, right, they fully migrated, and we have our spindle fibers that have attached to our homologous pairs, okay, to the centromere and our homologous pairs. So again, same thing that's happening during mitosis, during metaphase. So then we've got our anaphase. Anaphase is next. So let me draw my chromosomes again. Right, this time we have our 2 big ones, our 2 little ones, and we've got our centrioles on either side. Okay, so those have attached. So what happens during anaphase?
It pulls them to opposite poles.
Right, it's pulling our chromosomes to opposite poles. In this case, it's not the individual chromatids that get separated. Here is where we separate homologous pairs. So we are separating homologous pairs here. So with the separation of homologous pairs, okay, we have now gone from our 2N in anaphase 1 to now haploid, right, N. Because in each of our cells as we go on, we're going to have a fewer number of chromosomes, right? We go from having the 2 sets of chromosomes to having how many? We go from the two sets that we had here, right? Here we got the two sets, the one from mom, one from the dad, one from the mom, one from the dad. Now we only have one set in each of our chromosomes. So once we're here in telophase, again, the same stuff happens. Our centrioles start to break down, spindle fibers start to break down, nuclear envelope begins to kind of reform, but it doesn't form fully. Okay, so that's the end of meiosis 1. So after division, what stage should happen next? If after M phase, what should happen next? Not prophase. During it, we should have interphase, right? We should go back to our cell doing its normal function. Does that happen during meiosis?
No.
No. So once we go from meiosis 1, we go straight to meiosis 2. Okay, so meiosis 1 straight to meiosis 2. So we get no interphase. So no interphase happens during these two. So we go straight to meiosis 2. In meiosis 2, what we're doing here is we're reducing our total number of chromosomes. Because if we're looking in our telophase 1, so again, reducing the total number, right? Our pairs, right, they're doubled up, right? They're forming sister chromatids, so we have two copies attached to each other. So now we're taking our cells during meiosis 2, and we are separating the copies. So that we're left with, in the end, cells that only have one copy of each of the chromosomes. So let's go through and diagram what happens for all of these stages. So here we're in telophase 1. Let's go to prophase 1. So what are the events that happen during prophase? What's happening during prophase?
I forgot.
Our centrioles do what?
They start forming spindle fibers.
Right, they begin forming spindle fibers and start migrating. What happened to my nuclear envelope?
It's disappeared.
It's disappeared. It's gone. My DNA is still condensed because we just got done with division. Okay, so prophase 2, again, the same stuff that happens during prophase and mitosis, same things are happening here. And one thing to note, meiosis 2, it looks a lot like mitosis. The events that we're going to see once you diagram this out, it's going to look a lot like what happens during mitosis. It's going to look a lot like the video you guys made. So let's move in from prophase 2 to metaphase 2. All right, so in metaphase 2, right, we got our chromosomes still, but what are they going to start doing? They're going to line up on the midline or metaphase plate of our cell. So I did a bad job drawing this one. But we've got our chromosomes that are going to line up on each side, and our spindle fibers are going to attach. Okay, so we're still migrating them, moving them where they need to go. Okay. All right, so we're still attached. Everything's lined up on the metaphase plate down the middle. Okay, this weird looking cell right there, but there we go. Okay, so metaphase 2, we're moving into anaphase. Okay, so what's happening during anaphase during division? They're getting pulled apart, right? So our chromosomes are going to start separating, right? We're going to get them pulled to opposite ends of our cell. Okay, so here during anaphase 2, this is where we separate sister chromatids. Okay, so we separate our sister chromatids, so they become two separate things. Okay, so our spindle fibers are still here, centrioles are still here, and they're pulling them to opposite poles. So again, same stuff that's happening during mitosis. Okay. So we go to telophase, we got cytokinesis happening. That's going down. Okay, so what's happening to our chromosomes now? So we start to reform our nuclear envelope, so that's going to show up around our cells. What else is happening during telophase? Our DNA starts to do what? Start to unravel, right? Our DNA is already replicated. We don't want it to replicate anymore. We don't need extra copies. So we've got now our cells starting to separate, cleavage furrows are forming, and in the end, how many daughter cells did we make? How many are here?
4.
We have 4 cells, 4 daughter cells. Are they genetically the same or are they genetically different?
The same.
So let me draw them all out. If we're looking at our chromosomes,
And that's it.
they are going to be not the same, right? We've got different sets from different parents. So if we look back here we had 4 chromosomes in our cells, right? These are doubled, okay, because they form these sister chromatids, but we have 4 total, right? 2 that came from our dad and 2 that came from the mom. If we look down here, how many chromosomes are left in each of our cells?
One. Right?
We only have 2 chromosomes, right? We've got one in this case that came from the mom and one that came from the dad. So we are going for where our cells had 4, okay, 4 total, still 4 chromosomes, down to where we only have 2. So we go from our diploid number, which was 4, to a haploid number that is, what's our haploid number? 2. Okay. So we're reducing the number, and then do all of these cells have the same chromosomes in them?
No.
No, they're different. So these are all genetically different. Okay. And that genetic difference comes from 2 places. We can get it from here in prophase, where these things can end up joining together. So I showed you this yesterday, where they can form this structure called a, let's see. So we form this structure right here called a tetrad, right? And with tetrads, you can get crossing over, where we exchange genetic information. And we'll talk more about crossing over next semester when we dive into genetics. The other place where we can get variation is during metaphase 1. And that's just all dependent upon where these things lined up. So if we look at my drawing here, I had a blue one and the red one that matched this parent, but these switched around. All right, this just happened so that this red one lined up under the blue one and this blue one lined up under the red one. So when they got pulled, what happened?
The chromosomes got separated.
Right, they got separated, so then the set from the mom and the set from the dad got split and put into 2 different cells. Okay, so we're going to split the number of chromosomes. So they end up all being genetically different. Okay. All right, so do you guys have any questions about this?
Yeah, I can read that.
Mhm.